So we mentioned that mitosis and meiosis are similar in some regards. They both start with a diploid or a 2N cell. They duplicate in interphase. The big difference is the products. So the product of mitosis, 2 genetically identical diploid cells. Meiosis, 4 genetically unique or different haploid cells. This variation that we talked about in meiosis comes from recombination or crossing over and what we call independent assortment. And we'll look at that more later. It's going to add variation based on simply how the homologous chromosomes align in metaphase 1 and then how they align again and how things get separated in metaphase 2. This is much more important in genetics and evolution, and we'll look at that more later on to give you a sense of how that works. When we look at differences in chromosomes, I mentioned to you this word karyotype before, and it's essentially a picture of your chromosomes. So when we look at this picture of your chromosomes, what are we looking for? Well, most of the time we are just looking to make sure that the homologous chromosomes are pairing up correctly, so you get X's, that the proper chromosome number is there, and that the proper chromosome structure is there. To do this, it's very, very simple actually. All you need is a blood sample, and you put the blood cells into a hypotonic solution. Do you remember what that meant? You should. And then we look at essentially freezing the cells or doing kind of a still animation of these cells, and you can get a picture of the chromosomes that looks like this. Now, your chromosomes are not colored. These are colored with fluorescent tags in the lab to help look for chromosome abnormalities. So the idea is between the homologous chromosomes, so if this is one from mom and one from dad that is now part of you, we should still see the same banding pattern even though the alleles might be different. Even though mom might say brown hair and dad says blonde hair, the banding patterns would still be the same. In this case, the sex chromosomes show us an XY. So is this male or female? We also look for problems with chromosome number associated with these karyotypes. Trisomy 21, tri is 3. So trisomy 21 focuses on chromosome number 21, and it involves having 3 copies of that chromosome inherited. This is actually the most common chromosome abnormality that still results in a successful birth. We see a lot of chromosome abnormalities where the individual is never born. So this actually leads to the common disease name of Down syndrome. Down syndrome has a very specific set of symptoms associated with it, but the expression of those symptoms is very, very different because remember, chromosomes are full of genes. Genes code proteins, and all of these symptoms are based on a concentration of proteins. So depending on the concentration, the levels of those proteins, the symptoms or these characteristics might be highly variable. And we'll talk more about that later on as well. We found out recently that as mom gets older, the incidence of trisomy 21 increases significantly. Here's a karyotype of trisomy 21. You can see there are very clearly 3 copies of that chromosome. In this case, male or female. So looking at trisomy 21 as a function of mom's age of mom on the X, Number of infants with Down syndrome, 1,000 births on the Y. So you see we kind of cruise along where the incidence isn't very great, and then it starts to grow exponentially as mom gets older. And we think a lot of this has to do with complications associated with older eggs for mom and a process known as non-disjunction. So this is where during anaphase, the chromosomes fail to separate properly. So if they don't separate properly, in meiosis 1 here, we have a homologous pair that did not separate as it should. You end up with more chromosomes than you're supposed to in 2 of the cells and less chromosomes than you're supposed to in 1 and 2 of the other cells. So this would result in abnormal gametes, and so that would mean abnormal sperm or eggs, depending on if you're male or female. And then when we would take one of these sperm, so say you take this sperm with 3 chromosomes in this case, and you're going to put it together with mom's, who in this case only would have 2 chromosomes, so 2 plus 3 gives you 5. You're supposed to have 4. So this leads to great complications. In this case, 2 plus 1 gives you 3. You still don't have the 4 that you were supposed to have. Again, some complications could arise. Potentially, the child would not even be born. Non-disjunction can also occur in meiosis 2, so the sister chromatids don't separate here properly. And you see here we actually get some of the gametes that are normal, and then you would end up with several that are not normal as well. Errors in meiosis are not necessarily the worst thing ever. We look at them in humans as being significantly impactful on human health, particularly in offspring as the parents get older or are exposed to environmental conditions and so on. But what we do look at is that non-disjunction idea has actually led to some very unique species. Polyploid is extremely common in plant species and leads to hybrid plants and all kinds of very unique varieties of plants. We see it less frequently in animals, although I give you an example here of the gray tree frog. That instead of being a 2N organism as we would expect, it actually is 4N. That means that it has 4 copies of each chromosome. And obviously it's living and doing quite well. It's learned to live with that and adapt to that situation, but we see this very rarely in animals.